Okay, class, welcome to Chapter 2. I hope Chapter 1 wasn't too tedious and off-putting. Like I said, it is kind of an introductory chapter where we go over a lot of information very broadly, but now we're going to start digging into the specifics. And so in Chapter 2, we're going to talk a little bit about how the cells that start off as a ball of pluripotent cells that are able to become anything, these cells can become bone cells or they can become nerve cells, etc. How they come to the decision and how they start forming an identity and specifying identity. So we can kind of think of cells as the grains of sand in this octopus structure here. This octopus sculpture here. So each one of these individual cells, if looked by themselves, you would just see a grain of sand, but then when you take them together, they form structures. And we're going to look at how, throughout this course, how these structures are created and how cells know their purpose of what type of eventual final product, like a bone cell or a nerve cell or a muscle cell, they are to be. So differentiation is a process in which cells cease to divide and they develop a specialized structural element that gives them a distinct function within the organism. And so this is the last stage of a large cascade of events within the developing embryo. So these are kind of like the finalized form cells of the organism. And so some examples here is keratinocyte, which is a differentiated cell that produces keratin, and you might know keratin on your own body. Keratin is the components of hair and fingernails, etc., but also in other animals, hooves and horns and rhino horn. You hear about that unfortunate set of circumstances in Africa where it's being hunted for Eastern medicine purposes. So these cells, they differentiate into keratinocyte. They produce keratin, and then they have some sort of specialized function. And so we have some other examples here. Melanocyte produces melanin, which is the pigmentation in your skin, which I'm extremely lacking. However, those of you that are able to get tan, the sun will stimulate these melanocytes and produce more melanin, and then you have this nice golden tan. I'm not bitter, I'm just jealous. So you also have neurons, which are obviously part of the nervous system that create things like serotonin, which are part of the neuroreceptor response. So all these, we're not going to get too terribly into all these different cell types. These are just examples of differentiated cells. So now we're going to hit on a little bit of lingo. So commitment of a cell is the process of specification and determination. Now, specification is the fate of the cell or tissue is capable of differentiating autonomously, so it can differentiate by itself when placed in a neutral environment. So these cells will continue to differentiate or go down their path if you put them by themselves. However, their fate is not sealed yet. So if you would take those cells and move them into a environment where they have different neighbor cells that are becoming a different tissue type, they will adjust themselves. So they're starting down the path to becoming one of those final melanocyte or whatever type of finalized cell product, but they're not locked in yet. Now, determination is these cells are capable of differentiating autonomously. So that means they're able to, you take them out of the organism and they'll continue down their road to becoming whatever cell type. But if you place them with new neighbors, so say you take melanocytes and you place them near cells that are going to become nerve tissue or nerve cells, they will not adjust to their neighbors. They are already committed. They are going to become those melanocytes, and their fate is sealed. And then once you get beyond determination, you get the final product, which is differentiation. So this is kind of like starting down the path where specification is kind of the first step towards becoming a differentiated cell. Then determination is I've committed to this and I'm going to continue. And then differentiation is the final product. Okay, so here we have an example of specification versus determination. So this is just kind of illustrating what we just went over in the last slide. So here we have a ball of cells, and in it we have two different cell types. We have purple, which is muscle, and then we have yellow, which is neuron cells. So those are specified to become muscle and neuron respectively. So if we take out the muscle cell and put it in a neutral environment by itself, that cell has already started down the path to differentiating and becoming muscle. So it will continue and it will become muscle tissue. The same thing can be said for the neuron tissue. If we take a neuron cell out of this ball that's been specified, that cell will develop fully by itself in a neutral environment into a neuron cell, a fully differentiated neuron cell. Now, because specification can be reversed, we're going to look at it as in the context of a community. So if we take a muscle cell and we place it in a community of neighboring specified neuron cells, because it's specified and it's not determined yet, that cell will change and it will become a neuron based on its neighbors. So this is kind of a mechanism where these cells can realize, oops, I'm in the wrong spot. I'm not supposed to be with these neuron cells. A piece of muscle developing in the middle of neuron cells might not be good for the organism. So it allows this kind of flexibility to change to become what your neighbors are. But if it's determined already and specification is irreversible, then that cell will continue. And as you can see here, we have a muscle cell in the middle of all these neurons. So if it proceeds too far down the path and it goes from specification to determination, then at that point, that cell can no longer reverse back to a neuron and instead stays as a muscle cell with neuron cells surrounding. So how do these cells know what type of specification they need to pursue, right? So they start to figure out, okay, I need to become a nerve cell eventually, or I need to become a bone cell eventually. So we're going to really get into the weeds about how this happens, but the first thing we're going to start off with is this autonomous specification. So the fate of the cells are predetermined based on their position within the egg cytoplasm. And so the position they are when they're developing gives information to the cells as to what they should become. And so within the egg, there are these morphological determinants, which are like transcription factors, RNA, microRNAs, all these little things that are already in the egg cell when the egg becomes fertilized that will help to specify these early components as to polarity and what side of the cell they're on, the cells are on, their location, so they start to get that information as to what they should develop into. And so these cells know very early on what they're destined to become. So what's very interesting about this is that once these cells start down specification, as we've kind of showed in a couple of the previous slides, we're really going to hammer it home, is that even if you isolate these cells and they've started down that specification pathway, then they are kind of started their commitment, and unless they have neighbors that are telling them otherwise, they'll continue to become what they were specified to become. So in this example here, we have a ball of cells, a trochoblast, that is starting to specify. And as you can see, we talked about in the first lecture, I believe, that the mitotic spindles are one set of information that allows positional information for these cells. And so at this point, these cells will divide, and as you can see here, all the pink cells have developed cilia. So they've got a specialized function. So cilia are used in microorganisms for movement and motility. And so you can see it's starting to develop in part C of this figure here. Now, if we were to isolate those, take the pink part out of this blue bundle of cells and allow it to divide by itself, because of specification, it will continue down its developmental pathway and you'll still get cells isolated that develop cilia, and that same structure is preserved because it's started down that plan. Now remember, with specification versus determination, if we were to take part D here and put them with new neighboring cells that are specified to be something else, then they can still change their stripe, so to speak, and become a different type of tissue. So remember, specification, that is where you are destined for something, but you haven't fully committed, and determination is you fully committed to becoming whatever cell type, and your neighbors have no influence anymore. So as we talked about in the previous chapter, Conklin, the developmental biologist, noticed that some of these cells have a noticeable characteristic in C-squirts, that this cytoplasm has kind of this coloration to it. And like I said in the last chapter, this isn't preserved across all things, and so it's not this easy in humans and higher organisms. But within the C-squirt, he noticed this orangish dark yellow component, and that if you watched through development, that component always became the muscle of the C-squirt. And so, as you can see, my video is covering it, but you should have it in your slides, that around the edges here, the muscle, or the trunk, the core of the C-squirt, is still stained yellow. So these are morphological determinants that we talked about previously that are telling that positional information to those cells that they need to become those trunk muscles of this developing C-squirt. So to confirm this link between that yellow cytoplasm product and this autonomous specification that we're talking about, Conklin removed the cells from the B4-1 layer here of the C-squirt tissue, and looked to see how that affected the development of the C-squirt. And what Conklin found was that if you do remove those cells, then this tissue here that's highlighted or stained in red, the muscle tissue, is no longer there. And so from that we know that those cells were predestined to become this muscle tissue, and that since you remove them, the other cells aren't going to make up for that, right? They're going to just not be there, and that structure is not going to develop because those cells were missing. So using this technique, we can go back and create fate maps. We talked about fate maps as kind of this idea that we know what a cell is going to turn into based on its position, and we can kind of predict what areas of an embryo are going to develop from a blastocyte based on where they're located. And so by removing certain components at different cell stages, we can kind of see what those pluripotent cells, what we think are pluripotent cells, will eventually become. And so as you can see here, we have the 2, 4, 8, 16, 32, and 64 cell stage, cell stage. And when you remove cells, so we saw a couple slides ago, there's that cell that had, or that image of a developing blastocyte that had kind of this 3D structure, and there's a numbering concept to it where you have A's and B's, and then 4.1, etc., are locating the cells within the developing blastocyte. So if you remove one of those, for example A3, then A3 at the 4 cell stage develops into all of this: epidermis, endoderm, notochord, the brain, etc. And the other half, the B section, so B3, develops into also endoderm, mesenchyme, muscle, all these other different cell types. If you wait till a later cell stage and only remove, for example, A6.1 here, then all you are losing are the endoderm. So as the cell progresses, it becomes more and more specialized through this differentiation and specification process. So here's an example of that blastocyte that we talked about. So as you can see, it's numbered A 4.1, 4.2, B 4.1, 4.2 based on anterior and posterior. You also have the animal pole and the vegetal pole. So these are kind of differentiating the axes. And so if you are to remove some of these groups of cells at this stage, for example, if we remove B.41 and A4.2, or sorry, B4.2 and A4.2, just this section here, and we put them in a neutral area, so they're not surrounded by other cell tissues, then those cells will continue to develop based on what they are specified to do. So they will still become ectoderm by themselves. Now, if you can compare that to isolating just A4.1 down here, those cells will then develop and become different from A4.2. They will become notochord tissue because that's what they were specified to do within this developing blastocyte. So the specification process, if you remove cells, remember, and put them in a neutral area, so no influence from other cells around them or different cell types or different specified cell types, then they will continue their development. And this is how we can do that fate mapping to see what areas of this developing set of cells is going to be differentiated into. So what is it in that yellow substance that's in the cytoplasm of the cell that causes this positional information? And so an experiment was done where they extracted that yellow substance from the cytoplasm of the cell, and they implanted it into cells in different locations within this developing blastocyte. And so they wanted to see if by doing this, they could cause a change in the autonomous specification of those other cells. And it turns out that within that yellow cytoplasm containing substance, there's an mRNA that it's kind of a cheeky name. They call it macho. You know, it specifies muscle tissue, so obviously it's, you know, macho for doing that. And so this is a mRNA that encodes a muscle-specific transcription factor. So this little transcription factor or protein tells only muscle genes to turn on, right? So that is the component of this yellow substance that tells it to be, or this autonomous specification process, to become muscle tissue. Now, if we are to manipulate macho, we can see different fates of these cells that will not or will develop into muscle tissue. So in our control group here, you see that these are stained to show all the cells that contain macho. So we have 7 cells here that are expressing macho. Now, if we knock down macho through RNAi or knocking out the gene, you see that there's only 1 cell here that's expressing macho, and there'll be less muscle tissue that develop because of this. Now, if we overexpress macho or add them to other cells, then we see a lot of these cells are going to develop into muscle tissue, and you'll have more than expected compared to the control group. So this kind of cemented the evidence that macho, this mRNA that encodes for a transcription factor in the egg, is the determinant that tells the specification of these cells should become muscle tissue. So we just covered autonomous specification, where a cell is set on its path and will continue to meet its desired outcome by itself, right, autonomously. So we also have conditional specification. So that's where cells meet their respective fates by interacting with other cells, so knowing their neighbors. And some of the things, these interactions with their respective neighbors, include juxtacrine factors, which are cell-to-cell contacts. We'll kind of get into those. They're proteins that reach across cells. Paracrine factors, which are things that are excreted into the local environment, so stuff that's put out by the cells and the neighbors can sense it. And mechanical stress, so physical forces on the cells can cause this conditional specification. So if you're to move these cells from one location within the developing embryo to another, their fates change depending on their new location. So in cells that undergo conditional specification, we can look at that same transplant type procedure that we talked about earlier in the last chapter. And if we were to take cells from the normal back developing area of a blastula, and we are to transport them to the kind of stomach or ventral region of the developing blastula, we see that those cells that were originally going to develop into the back now develop to the ventral or the stomach side of this organism. So because they are sensing their environment, conditional specification cells will look at what's around them and adjust their strategy as to what they're going to develop into depending on where they are within the cell. So as opposed to autonomous specification, where they're going to develop, if you isolate them, they're going to develop fully into whatever tissue they started out as. Conditional specification, they look at the neighbors and they decide, okay, I'm going to change my strategy. What's also interesting with conditional specification is that if you are to remove cells from a very early developing blastula, then those nearby cells are able to fill in the gaps as long as they're the same tissue type and adjust for these missing cells. And thus you end up with an organism that looks like if you were not to have removed cells. So they can kind of fill in the gaps and continue proper development even if some of these cells are removed. The developmental biologist Weissman proposed a theory called the germplasm theory. And this is an example how not everything proposed in science holds up over time and that our understanding of science changes over time. But he proposed that each cell of the embryo would develop autonomously. So he proposed that autonomous specification was the way that all cells within the developing embryo would perform. And so he proposed in this theory that egg and sperm provided equal chromosomal contributions, which is correct. So you get 1/2 of your chromosomes from your mom and 1/2 from your dad, that's correct. And that they carried the inherited potentials for the new organism. So he proposed that those chromosomes are what pass down the genetic information, and that was also correct. However, he theorized that the chromosomes broke into chunks that were unique to each cell type. So if you're going to be a skin cell, then you would only inherit the chunk of the chromosome that had the genes for skin cells. And that if you were a melanocyte, you would inherit only the chunk of the chromosome that had the genes for melanocyte specification. And so that only gametes, only the sex cells, had full complements of your entire genome, all the chromosomes. And that's incorrect, we know now. So each cell has every copy of every gene, and they're only turned on and off specifically for each cell type. So a disciple of Weissman named Roux attempted to demonstrate that this autonomous specification theory was correct. And so in his experiment, what he did is at the 2-cell stage, he used a hot needle to destroy one of the cells. So now we have half the starting material of a developing embryo. And from this, he found that only half of the embryo fully developed. So as you can see here, we start to see kind of the neural fold and development of the notochord, and that the destroyed half never really developed into anything. And so this gave credence to the idea that autonomous specification that Weissman had predicted was actually correct. So Hans Dreich, another developmental biologist, did another set of experiments to test this autonomous theory. And what he did is he removed the fertilization envelope from around the developing cells and shook them gently to break them apart. He later repeated this experiment using calcium-free seawater to break apart these 4 cells. And so at this stage, he then allowed the 4 cells, to develop individually. And he found that each one of those 4 cells developed fully into the organism. Now, if Weissman's autonomous theory was correct, then we would expect each one of those 4 cells to develop into 1/4 of the organism. However, that was not observed, and that's not the case. And so this shows that the autonomous development is a strategy that cells undergo in addition to this conditional specification where your neighbors play a role. So in some instances, autonomous specification takes place, and in some instances, conditional specification takes place. Dreich also did a second set of experiments to confront Weissman's autonomous theory, and he called these set of experiments the pressure plate experiments. And so Dreich thought if Weissman was correct in his idea that different chunks of the chromosomes go into different cells and tell them what to become, like skin cells, etc., that if you were to reshuffle those cells, then you should have an organism that's all mixed up, where skin's on the inside and the heart's on the outside, and all sorts of different mixture of cell types, as opposed to the normal structure and development that we see. And so what he did is he took a developing early stage developing embryo, and he put it between two glass plates, as you can see here. And under this pressure, because this blastocyte that's developing is a three-dimensional organism or structure, if you put it between plates, when those cells divide, they're no longer able to divide. They can only divide in two directions, not in three directions. So this causes those cells to still divide, but kind of reorient. And as you can see here, in the normal cleavage of these developing cells, we see kind of this preserved structure where we have 1A, 1B, 3A, 3B, 5A, 5B, 7A, 7B on the inside. But then if we look at how it developed underneath these pressure plates, we have 1A, 2A, 3A, 4A, 5A, 6A, 7A, and all the B's are on the outside. And so this is a reshuffling of how the orientation and the neighbors of this very, very early developing blastocyte. And so what he found is that if you were to do this, again, in Weissman's theory, you should have a weird organism where all these different tissue types are mixed up and not in the right place, because the normal position for the cell containing the skin part of the chromosomes would be on the outside, and now it's on the inside. However, that's not what we observe. Instead, this cell, or this animal, developed normally. And so this shows that at the very beginning, this autonomous theory would propose that you are destined your cell type from the very beginning, because those chromosome chunks for each different cell type are allocated to their cells, but this isn't the case. Instead, at this early stage, these cells retain their pluripotency and are able to become any cell type. So the last type of positional or specification that we're going to talk about is syncytial specification. And this occurs in insects. This is where a cytoplasm has a lot of nuclei within it, called syncytium. Specification of these cells are called syncytial specification. And this, like I said, in insects, so Drosophila melanogaster, which is the fruit fly, is one of the organisms that undergo this type of specification. And in this, there are 13 nuclear divisions, so the nucleus replicates itself through 13 times before there's any cytoplasmic cleavage. So within the cell, you are gaining more and more and more nuclei before there's any sort of cell division itself. So in the developing Drosophila egg, we have all these different blue dots. These are all independent nuclei. If they're blue, they're not actively dividing. And if they are purple, they are actively dividing nuclei. Now, as you can see, this kind of proceeds in a wave from one pole of the egg to the other pole of the egg. And so within this egg, there's a gradient of different factors, and we're going to get more into them in further chapters, but there's a gradient that kind of tells each nuclei what position they are within the egg, whether they're at one pole where there's a high concentration of this morphological factor, or the other pole where there's a very low concentration of this morphological factor. And these morphological factors determine the cell fate. So what happens is these different nuclei will line up along the outside of this egg, and they will build cellular walls around them. So they do this process kind of like a budding process where the nuclei are all replicated first, and then they're kind of allocated out into these different to the sides, and the cells form along the sides, as opposed to mammalian development where you have one cell, it becomes two, and then two become four, etc. This is kind of a different strategy that's unique to the syncytial specification. And so once they are formed into cells along the outside of the egg, they use those gradients that exist within this developing egg to determine their relative location and what type of cell that they're going to develop into. And so if you're on a high gradient side, for example, you will have a lot of those morphological factors, and that might tell those cells, okay, you're going to develop into the head of the organism, while a low concentration of those morphological factors on the other side would lead it to develop into the thorax or the butt of the fly, developing fly. So what prevents these nuclei from just kind of floating off into oblivion? How do they get to the outside of the cell where they are packaged up into individual cells later? Well, these nuclei have kind of this actin cytoskeleton network where after between cell divisions, they put out this dynamic microtubule network that consists of actin and microtubules, and as well as these motor proteins. And what they do is they form this kind of web type scaffolding within the cell, and they're able to migrate using the scaffolding to the outer edges and maintain them in the proper position for those cell membranes to be constructed around those nuclei. And so that's kind of how this packaging process of these nuclei happen in the syncytial specification. So we mentioned this gradient that exists across this egg, this developing egg, and in this case we're going to talk about one that you're going to hear a lot about throughout the semester, and it's called bicoid. And bicoid is a morphological factor that establishes the anterior-posterior gradient across the developing embryo. And the highest concentration is at the anterior. So you can see here it's stained with like a GFP or green fluorescent protein, and you can see that it's very bright at the anterior position. And as you proceed down to the posterior, you see that it is much less bright. So there's a reduction in the concentration of this as you proceed across the egg. And so this gives one aspect of positional information. There's a lot of gradients that overlie on top of each other, and we'll talk more about them as we proceed through the class. But this is kind of a big key player in early development of establishing the anterior-posterior position of this developmental process. So we can graphically represent this gradient of bicoid across the organism. So we see the anterior-posterior distance here on the bottom, and we see that at the very anterior end we have a very, very high concentration of bicoid, and as you proceed to the posterior end we have a much lower concentration of bicoid. Now the question becomes, okay, once you get to about 0.6 away from the anterior position, this level of bicoid is pretty similar, right? So, if you look at an organism, the cells that become the very tip of the thorax compared to a little ways up the thorax, they're different, right? And so, for example, if you're a bee instead of Drosophila, you have a stinger at the very end of the organism, but that's a different organ or different structure than something that's a little bit more anterior from that. And so this only gives us one part of the equation as to how we determine these different gradients, or these different positions with gradients. And so as we'll see in the next slide, we have other gradients that proceed in the opposite direction. And there's actually a lot more that we're going to overlay all of this, but another one is called caudal. And I apologize for trying to draw this with a mouse because my handwriting doesn't look very good. But caudal is a gradient that proceeds in the opposite direction. So a stinger for example, in the development of a bee, would have to have a very high amount of caudal and a very low amount of bicoid to develop. And so that differentiates it from cells that are further up, which has a medium amount of caudal, but still this low amount of bicoid. So this gives more and more detailed information as to the position of these cells as they go through this developmental process. So as I alluded to in the previous slide, we have the second gradient, caudal, which starts at the posterior where it's the highest, and it tails off towards the anterior. So if you see, we can overlay these two different gradients and get more detailed information, where if you have a extremely high amount of caudal and a extremely low amount of bicoid, you're the very posterior. But if you have a little bit of caudal, a little bit of caudal, bicoid, and a decent amount of caudal, then you're a little more anterior. If you have equal parts, caudal and bicoid, you're kind of the middle, etc. So they can kind of depict this with this changing color from red to kind of a purple to a blue, that these two gradients exist and overlie one another. And from this, this syncytial specification is occurring, you can see that we get a little bit of positional information where we say, okay, if you have very high bicoid and low caudal, you become the head. If you have a little bit of caudal but a lot of bicoid, you become the thorax. Conversely, if you have a little bit of bicoid and a lot of caudal, you're the abdomen. And if you have only caudal, you become the tail. So we talked about those three different types of specification. We have autonomous specification, conditional specification, and syncytial specification. Now, it's important to note that these predominate in most invertebrates. So mostly autonomous specification is in invertebrates like the sea squirt. There are cases that they will use both autonomous and conditional specification. In conditional specification, most vertebrates and a few invertebrates use conditional specification, which was depending on cell position. And then syncytial predominates in most insect classes. So this is just kind of a review of the three different types of specification that we went through. For your knowledge, kind of a summary slide as to how these three different modes of action are kind of grouped by organism. That is the end of this chapter, and I'll see you in chapter 3.